Hello, my name is Emily Worth, and this is my model of the operon that controls the production of compound B. An operon is a collection of genes controlled by a shared regulatory element. And operons are actually an example of pre-transcriptional regulation. The function of operons is to ensure that only the products that are needed are produced, which decreases the amount of energy needed and increases the efficiency. For instance, operons may turn on or off transcription, which allows the genes to either be turned on or off. For example, when transcription is turned on, the repressor is not attached to the operator and the gene is expressed. However, when transcription is turned off, the repressor is added to the operator and the genes are not expressed. There's actually two types of operators, or operons. There is the inducible operon, which is shown in compound B. And this means that transcription is normally turned off, shown here. And only under certain circumstances or environmental conditions does the transcription process occur, shown by the repressor leaving the site. And this increases the energy efficiency, meaning that the transcription process is not always occurring and not all the genes are expressed, which saves energy for the cell. However, there's also another type of operon called regulatory or repressive operons, and this means that the transcription process is normally turned on. However, during certain circumstances, the transcription process is turned off, shown by the repressor going to the operator. However, as I mentioned, this is an inducible operon. So I'm going to go through the main components of the operon before I go through the process. And so we're going to start at the promoter. And the promoter is a place where the RNA polymerase, described here or shown here, attaches to the DNA, and this starts the transcription process. Next is the operator, and this is the on or the off switch that determines whether transcription will or will not occur. Next are the genes, and I included 3 genes, gene 1, gene 2, and gene 3, shown in yellow. And the genes they participate in generating a specific code for compound B, shown by these yellow beads here. And finally, shown by the red, is the terminator. And this tells the RNA polymerase, after it's gone through the DNA sequence, to stop the transcription process from continuing to occur. So it's the ending point. And shown by this purple square, as I mentioned earlier, is a repressor. And this blocks the transcription process from occurring. Therefore, it's an example of negative regulation. A fun fact is that repressors are actually allosteric, which means that the binding site alters its ability to bind to DNA. Its shape may change, therefore. And an example includes when the repressor is added onto the operator and an inducer is attached. The shape of the repressor changes and it releases from the operator, which allows the process to continue. And this is the mRNA sequence shown here after the transcription process. And then this is compound B. So going through the process of the normal circumstances, this is the RNA polymerase, as I mentioned earlier. It attaches to the promoter. However, because transcription is normally turned off, the repressor is attached to the operator. And this prevents the RNA polymerase from continuing down the sequence and transcribing the DNA into mRNA. It blocks it. However, during certain environmental conditions or circumstances, the inducer is added, and this allows the repressor to release from the operator. And this allows the RNA polymerase to go through, transcribe the sequence. It stops at the terminator in red, and it transcribes the mRNA, which acts as like a framework or messenger to create compound B. And this is the process of the operon for compound B. And the importance of this operon is the fact that the gene is not always, not all the genes are expressed. The transcription process is not always occurring because compound B is energetically costly. And so it would waste or use a lot of energy if this process was constantly occurring. But because of the on and the off switch, this allows the genes to only sometimes be expressed, which allows for more efficiency. And this is a closer version, or a closer look at the components of my model. Thank you.